So it is there?
Yes, sir, we can see, sir.
Okay. So good evening, you all. And thank you, Karuna, for giving me this opportunity to address and to talk about our work to this audience. I will be presenting the introduction to molecular methods in diagnosis of fungal infection with MALDI TOF as an alternative. Although the whole molecular methods which are being used for the diagnosis of fungal infection, it's a vast topic. So I will try to summarize, I'll try to, brief, and I'll try to touch upon all the area during my talk. Now, as the audience is basically interested for mycology, and you know that in the world, about 1.5 million fungus exists out of this 72,000 species known. And it has been found that every year, around 1,700 cataloged, and 300 to 500 out of them, they are known for causing human infection. Now the question is, why do we need the molecular identification or advanced technique for identification? Recently we published a study that in a fungal keratitis, there are newer agents or newer fungi, they are causing fungal keratitis. Means the fungi, they are known for plant pathogen because of some reason, the elevated temperature in the world, more adaptation of the fungi. The plant pathogen, they are slowly becoming the human pathogen. And every day, our list is growing. So in that case, whatever the, the conventional method or the diagnosis procedure, we have to update. So there we need some better method for the identification or diagnosis. Now, in case of fungi, we know that fungus can exist in three forms. One is yeast. You know that Candida, Cryptococcus, they are the example. Molds like Aspergillus fusarium, and some of the fungi, they can be either form, like they are calling dimorphic. The best example is Histoplasma and Penicillium or Sporotrix. Now, if we talk about the fungal infection, although this is not the area of my talk, but we know that there are three main categories of the fungal infection, that is superficial or cutaneous infection, subcutaneous infection, and systemic infection. Now, when we have the fungal infection, our duty is to diagnose. So, we have the two ways we can diagnose. One is the conventional diagnosis, another is the molecular diagnosis. So, before I start molecular diagnosis, I'll just talk a little bit of conventional diagnosis, why we need the molecular diagnosis more. Now, conventional diagnosis, it depends on mainly the direct microscopy, histopathological diagnosis, and the culture, which which are known for the gold standard. Now, in this slide, you can see this is a weight mount from a tissue sample with KOH 10%. So ideally, that 10% KOH, it dissolves the tissue and the fungal body, come out, and under the microscope we can see. But in this, picture, this I'm showing that is the slide culture. Means in the conventional method, the fungus, it's not like a bacteria straightforward, whether it's a gram positive or gram negative, or the cocci or bacilli. Rather, for the fungal, conventional identification is more tricky. Means you have to understand their hyphal and conidial arrangement, their morphology. So that way, the slide culture is the best option, and we are doing this for the fungal identification. But for this, you need to have expertise. So one should be expert for the fungal identification, for the conventional. For the direct microscopy, there are some, fluorescent-based dye also introduced in the past. One of the examples is the calcofluor. So, the calcofluor, it is basically binds to the chitin, and the chitin is a specific property of fungi. So, if this fluorescence will bind with chitin, so we will not miss any fungi. So here it is the the hyphal form, and it is the yeast form, but still it has the drawback, like it can bind with the non-specific, and you may come up with some artifacts, which is it seems to you that it may be positive. So one should be careful. So this is another example of the direct microscopy. In case of Cryptococcus, we are using the Indian. So it's a very simple, and any a normal setup lab you can do it. So basically, what happened? This is a negative staining, and this outside the Cryptococcus cell wall there is a polysaccharide or hollow zone. It is created. So through the India ink, under the microscope, we can easily see it and diagnose it. So another I told before that it's a histopathology based diagnosis. So in the histopathology based diagnosis, we are getting the tissue sample, then we process it, and then we are doing the H&E staining, and we are diagnosing. But there is some difficulties, like it detects fungi, either yeasts or filamentous, or in the filamentous, whether it's a hyaline or pheohypomycetes. But it cannot tell us the identification, means whether it's a aspergillus or whether it is a fusarium. So that is the drawback. And lastly, we are relying on the conventional identification, that is the culture based. So you know that this is very, every lab is popular, that we get the patient sample, we process it, and we inoculate the sample to the certain media like SDA or PDA or BHIA, and after certain time, there is a growth. If that particular organism is there, so the particular organism will grow. And eventually, we go for the direct microscopy, we go for the slide culture, and we identify. So once we get the identification done, so it is a gold standard, means when you are getting something from the patient sample, so, nobody can question. But still, there are some drawbacks. Like some fungi, it takes, in case of fungi, it varies their growth. So some fungi like yeast, it grows overnight. But some fungi, it takes 1 week to grow. Some fungi, even slow grower, even take 4 weeks, like in case of certain pure hypomycetes group of fungi. But clinicians, they are always waiting for the identification as quick as possible, and the in vitro susceptibility, if possible. Then they can, treat the patient. So our aim is to treat the patient as soon as possible. Now, recently, some new conventional identification like Chromaker, it has been marketed. But on the basis of the color, we can differentiate different yeast colonies in the Chromaker, but still it takes minimum 24 hours. And there are, some cross reactivity and not clear phenomena we are facing. So the problem of conventional diagnosis is, in case of, I have already discussed the direct microscopic inspection of specimen, the sensitivity is poor, and it is difficult to get the deeper specimen. In case of culture, the recovery of the culture, in the culture is very, very less. Some non-sterile specimens, it's a problem. And another problem is the contamination. I can share one of my experience. We are working with the corneal scraping, a culture in our lab. So one of the students, she was coming with, sir, every second day or third day, I got the aspergillus. But when I checked slide carefully, the plates carefully, I found that this is not growth from the sea. This is the growth from the outside. So it might be contamination. So you have to be very, very particular and expert in case of culture-based diagnosis, but still it is I'm the gold standard until now. And later on, the last is the histologic diagnosis. It is difficult to go to the deeper tissue, and as I told before, that it cannot identify the fungi. So we need some alternative method to overcome these drawbacks or the difficulties. So in that case, the molecular diagnosis is important to know, and molecular diagnosis of fungal infections. Now I'll start this. So what is molecular diagnosis? Molecular diagnosis means the basis of the molecules. The molecules can be DNA, the molecules can be protein, the molecules can be anything like cellular component, like lipid, carbohydrate, anything. So anything, any molecule when we are using for the diagnosis, that's technique is coming under the molecular diagnosis. So now it is very, clear that one of the molecular diagnosis is nucleic acid based. So nucleic acid based diagnosis, we have PCR based detection, we have DNA probe based detection, and we have DNA sequencing. So PCR based detection can be either targeted gene or sequencing, and then real-time PCR based. And DNA probe-based technique like line probe assay, microtiter well hybridization, microarray in situ. But I'm not going to discuss all these DNA probe techniques, as some of the publications are there in the research purpose, but it is difficult to continue in our routine lab. So I will be focusing on whatever it is important for our routine lab, and what will ultimately help you if you know that I will touch upon. And lastly, definitely I will talk about the DNA sequencing. The second molecular diagnosis or molecule-based diagnosis is the biomarker-based. So biomarker-based, I told you that it can be any molecule which we can use as a biomarker, like cell wall component. In case of fungi, if we see the cell wall, we have 1,3-beta-D-glucan, we have mannan, we have galactomannan. So this can be used as a biomarker-based diagnosis. Capsular antigen, the cryptococcus, in case of cryptococcus, the polysaccharide or outer circle polysaccharide antigen can be used to detect the, cryptococcus infection. And lastly, the cytoplasmic antigen, that is enolase. Although there are some paper, but the use is not that many. And lastly, I will talk about the proteomic based, and I will talk the MALDI based identification. So now I'll start one by one. First, the nucleic acid based diagnostic techniques, or in the mycology. Now, you have to understand this diagnosis can be in two ways. One is directly from the patient sample, and another is we are isolating the culture and identifying the culture which we isolated from the patient. This identification of the culture or fungi, we can also diagnose. Now, when we are talking about the identification or diagnosis from the sample, so what would be the sample in case of fungal diagnostics or fungal diagnosis? It's blood and its components. It may be whole blood, or it may be the blood component like plasma, serum. The biopsy tissue, always we are looking for the fresh biopsy tissue. And recently, that in the COVID-associated mucormycosis cases, this fresh tissue of biopsy is always helpful. Now, FFP or formalin fixed tissue, we can also use for the molecular diagnostic. And lastly, the body fluids, different body fluids, including the ocular taps, like ocular taps or vitreous tap we can use. We can use bowel sample, CSF, ascitic fluid, pleural and peritoneal, pericardial or synovial fluid as a sample. Now, these are all from the direct sample from the patient. And as I have told, another way we can diagnose, that is from the sample you get culture, and finally you identify the culture by using molecular method. Like here I showed two pictures, one is the mold, another is the yeast. So this is the two way we can diagnose the fungal infection. Now, any DNA based or nucleic acid based technology starts with the DNA level. So as you all know that the DNA what we are using, this is actually it's a gene, and this DNA, it is existed in the chromosome, and chromosome it is in the nucleus, and this finally this gene, but different gene we are targeting for the molecular diagnosis. Now, as you all know that Watson and Crick, they explained the chemical structure of DNA in 1953, and from where we know that DNA is a double helix structure with 4 bases: adenine, thymine, guanine, and cytosine. Adenine and thymine, they are linked with the double hydrogen bond, but guanine and cytosine, they are linked with the 3 hydrogen bonds. So this is the simple the DNA structure, and this DNA we will use for the nucleic acid based molecular diagnostic. So it can be in 3 ways: PCR based detection method, and then as I have told, nucleic acid hybridized method, and DNA sequence based method. Now when we talk about the DNA based method, DNA can be extracted directly from the pure culture and from the patient sample. So first I will tell about the pure culture. So you have a pure culture, and you have to first, because you know from the previous slide that the DNA, it exists inside the nucleus, and in case of fungi, the problem is the fungi is having a very tough cell. So unless until you dissolve the cell wall or you break the cell wall, your DNA will not come out to the solution. So first thing, we have to break the cell wall. So the breaking the cell wall, it can be different way. Now it can be through the lysis buffer, or enzyme degradation, or it can be the mechanical degradation. Degradation like with the vortex, vigorous vortex, vortexing. So first step is to break your cell wall. So once the cell wall is broken, all the DNA comes out to the solution, and then you start all these steps. You then this solution mixed with the phenol, chloroform, and isoamyl alcohol, you vortex it. So whatever the impurities or attached molecules with DNA, like histone or any other protein, they will excluded from the DNA, and you will get the pure DNA. Now later on, we have to precipitate DNA, because DNA is a soluble. They are soluble in water, but you have to precipitate in the ethanol, and then after precipitation, we have the DNA. That DNA we have to quantify, and we have to quantify by using nanodrop, and we have to quantify with the 230 by 280 ratio. And finally, when we have the chromosomal DNA, if we run the gel, it will give you this kind of picture. So that way we are extracting DNA from the pure culture. And from this DNA, now it is, you can go anywhere you want, have one. So I'll come to the later on. But there is a little tricky, to use the patient sample while you are extracting DNA. Why? Because when we are doing the patient sample for DNA isolation, first thing, we have to dissolve the tissue if it is a tissue sample. Another thing is, if it is a blood sample, we have to lyse the red blood cells. So there are little more steps involved. So this is little older method where you can say this the tissue is lysed, then add alcohol. And in the old, the previous days, they used to, take this DNA fragment into the glass tube, and then the people purify the DNA. But recently, we have so many kits available in the market, like the hygiene, that is QIAMP tissue kit, gene releaser, this is all the kits available. You can use this. They have set the protocol. You can use this and you can extract the DNA. So now you have got the DNA, either it is a patient sample DNA or it is a pure culture DNA. Now, only DNA will not serve your purpose. You know that for the PCR, you have to have DNA template and you have to have prime. So now you have to understand the selection of the target gene for PCR. Now, in case of fungi, we are targeting the ribosomal DNA. Why? Because it is multiple copy. And in this ribosomal DNA, we have both conserved region and the variable region. So now there are so many terms you are coming to know, like ITS. This is internal transcribed spacer region. Like LSU, large subunit, SSU, small subunit. So just I am explaining this area. So if you understand this, this ribosomal DNA, it is very, it will help you to, design the primer and execute your experiment. So what is this? So this is the whole ribosomal DNA. This is ITS1 region. It is usually 208 plus minus 56 base pair. ITS2 region, this is 310 plus minus 67. And in between, this is 5.8S. And this is 18S SSU or small subunit and 28S or large subunit. So this is the whole region. And now, this ITS1 and ITS2, they are mostly variable region. So people are designing primer from this region. So if you are designing the primer from ITS1, so this is the arrow, this is the forward primer, and this is the arrow, this is from the small subunit, and this is from the 5.8 or subunit. So this is the reverse primer, and this is the forward primer. So you can get around 300 base pair product if you design properly from this area. Similarly, in the ITS2, also you can design the primer this way. Now, there is another term you come to know, that is Iga Spot or intergeneric sequence. This is a stretch of DNA sequence located in between all these ribosomal DNA. Now, in case of Malassezia, we use this Iga region. And if we stretch this large subunit or LSU, we will find there are two regions, D1 region and D2 region. So most of the primers in case of fungal diagnostics or identification, we design from here. So this is the large subunit, and only this D1, D2 we are using for designing the primer. Now, come to know, these different primers or pan-fungal primers, several labs people are using. This is ITS1, ITS2, ITS3, ITS4 for ITS2 region, NL1, NL4, this is D1, D2 region. So this is, you can get everywhere. Most of the publication you can get. But the problem is, there are, and ITS1 and ITS2, we are successfully using for the identification of fungus. Any, fungus, like if we want to identify up to species level, they are fantastic. But in some of the cases, we are not getting good result, or we are not getting the proper speciation. One example is the Fusarium. If you have Fusarium solari, if you have Fusarium oxysporum or incarnatum, whatever, the Fusarium ITS1, it will give you only the Fusarium species. It will unable to, resolve whether this is what species. So in that case, we are using either elongation factor, that is EF1 or EF2, or the RNA polymerase II gene, that is RPB2 or forward and reverse. So in certain cases, we have to think and we can change our plan. there are certain housekeeping genes also we are using for identification of fungi. I mean, most of them you have heard that beta tubulin, calmodulin, actin. So these are all the housekeeping gene also we are using for the diagnosis of fungal infection, or basically the identification. So these only problem with these genes are they are not the multiple copy, they are the single copy gene. And this particular diagram shows when we are designing the primer and identifying, you see IPS1, IPS2, LSU. So your sequence variability is going down. Similarly, the discriminatory power of the speciation is also going down if you come from ITS1 to ITS2 to 5.8S to LSU to SSU. So I hope I could be able to, tell you how we are designing primer, how we are targeting for the PCR. Now, we have targeted the sequence and we have, designed the primer. Now we will do the PCR. Now this PCR can be different types. The PCR can be targeted single gene, which is also called monoplex. The PCR can be duplex, means you can target two species together or two genus together, or you can target multiplex. Like one of our case, study, we are doing 5 different genus like Aspergillus, then Fusarium, then Stegosporium, Alternaria and Carbolarea. They are mostly causing fungal keratitis. So we are standardizing one multiplex by using these 5 sets of different genus primers. So another is the nested PCR. When some of the PCR, they are not really giving you the good result in one step. So initially we are using long segment, and from this long segment, the product we are using again at the small segment. So that way we are calling it nested. And lastly, real time, which I will talk to you later. So this is the simple PCR process. I think most of you know that we have to use the PCR. We have to first make the PCR mix or the PCR mix or the PCR reaction. Like we have the buffer, we have DNTPase, magnesium chloride, we have primer, we have DNA polymerase, and finally we have the target DNA. And after we mixing it, we put it into the thermocycler. And in the thermocycler, we have 3 cycles, that is denaturation, primer annealing, and extension. And this cycles, it also varies from 25 to 40 for the proper PCR. And this is the PCR machine. You put this, you program, and you just leave it. It will take 1 to 2 hours or 3 hours, depending upon your program. And then when you have PCR, actually PCR, what? PCR is nothing but to amplify of your certain product from 1 to 1,000 or million. So this is the way we are doing the PCR, and your PCR is successful or not, how you will ensure. Finally, you run the product in a gel, the agarose gel. So in the gel, you have to run one ladder or the molecular marker, through which you will come to know this the band size and your product size you can match. So there should be one positive control, one negative control, and your sample. So this is the way we are doing the PCR. So there are certain other variation, other PCR which I am not talking, but in case of PCR also, different kinds of PCR you will come to know. So once you have done your PCR, now there is a question that whether if you have this band, and this band we have amplified with a specific Aspergillus primer and Aspergillus specific DNA. Now, how I would know that this is the proper amplification or not, any non-specific? Now, finally, we have to do the sequencing. And we have to do the sequencing. So sequencing, it is previously it used to be the Maxim Gilbert method. It was very tedious and cumbersome. But lately, in I think 1977, Sanger, he introduced this chain termination method, which is, everywhere this process is using. So in case of fungal identification or diagnosis, finally we are doing the sequencing. So what we are doing, if we talk about in short, first we have clinical specimen or we have the pure culture. We extract the DNA from there. That we it will subject to the PCR, and then the PCR, it will be sequence, and this sequence will match with the different database, and we will come to know what species it is and what fungus it is. This is simple. When we do the sequencing, we are getting this kind of peak, and you see there are different color, like A is different color, C is different color, G and T, they all are different color. So what we are doing, this is this is a different software. We are opening this our sequencing result into a software, then we copy it and we put it to another software where you will get only the sequence like ATGC and so on, and then you can match. So in case of PCR sequencing, we have the clinical specimen, we extract DNA, we design primer, we then sequence, and we match the database. So what are the known database where we match our sequence? Like this is the EMBL, it is UK based. GenBank, you mostly known and by NCBI. Then Bionomics, it is a CBS site. You can try this. Then International European RNA Database. And there are some other also. Recently, our ISHAM, they have introduced the IPS DNA barcode database. That also you can check. So this is the way we are checking out the database. We are matching our sequenced data, and we come to know the conclusion or the final diagnosis. Now, there are some pitfalls also for the PCR reaction. Like PCR reaction, there is a contamination, because if you don't follow the proper procedure, don't take the precaution, and if your DNA gets contaminated, you will get the false result. So DNA contamination, it is a very, very important. Who are working with this PCR-based diagnosis, they should be aware how to avoid the contamination. Now, when you are identifying any organism, either it is from the sample or from the culture, and I am sequenced it, you don't know whether it is a colonizer or whether it is actual pathogen. So this is the drawback, and we cannot do the susceptibility testing. But I will only focus on the contamination part. Like I can share one of the incidents in our lab. A couple of months ago, we are getting, whatever we sequence, we are getting Candida. We are sequencing Aspergillus, we are sequencing Mucor, we are getting Candida. Finally, we found there is particular reagent. It was contaminated with the Candida primer, and then Candida DNA, or we are getting all the Candida. So then finally we sort it out, we resolve the issue, and then we are fine. So that way you have to be very careful. And this is the real-time PCR now. I thought in the COVID era, everybody knows that real-time PCR. Everywhere, most of the labs, we have the real-time PCR. So just one or two slides, and really we are trying the fungal diagnostics by using real-time, as we have most of the labs the real-time PCR. So this is a nice scope for us to work on this. So we have two real-time PCR. One is the Biorad CFX96 well, and the Roche cycler, a light cycler. Now, why do we do the real-time PCR? Because in case of PCR and the sequencing, we cannot, that is the quantity. We do the qualitative detection, and we do the detection of copy number, and then relative and absolute quantification of the target genes. Like if we want to, see the expression level or gene expression, or we want to determine the microbial load, then we want to do the genotyping, SNP analysis. All the cases we can use this real-time PCR. So we have to design our experiment and execute. Now, in the real-time PCR, there are two types. One is we are using cyberbin, and another is using Acman probe. So cyberbin, it is very simple. Like whenever there will be a double strand, that your fluorescent dye will attach, and then it will give you the measurement. But in case of Acman probe, now there is a probe, and in this probe there is a fluoropore and there is a quencher. So and there is a primer also. So when the primer start sequencing, and during that time, this reporter gene cut from this probe, and this fluorescent dye, it will release in the solution, and that's we are measuring, and we are doing the CT value. Now, in both of cases, some of the advantages or disadvantages, like in case of cyberbin, this is a simple assay. This is more sensitive, no recruitment of designing level probes, less assay time, and this is with low cost. But there are some disadvantages, like dye will bind with it, like in case of, and that is the main drawbacks. Like when we are doing the PCR, sometimes we get the primer dimer. So primer dimer, it is also double stranded. So you may get the fluorescence. You may get the fluorescence, and you may get the high CT value. It doesn't mean that that is the real. So that's the drawback. And another drawback is you cannot do the multiplexing. Means I was talking about multiplexing, the normal PCR. And in the real time also you can do multiplexing, but you have to use the Acman, not the cyberbin. And the melting curve analysis required to exclude the off-target application. Similarly, the Acman also, it has some advantages and some disadvantages. So this is the whole about molecular or nucleic acid-based techniques. Now coming to the biomarker-based techniques. Now, what would be the ideal biomarker? Ideal biomarker, it should be able to clarify or reflect early stage of diagnosis. The marker must be easily detected without any complicated medical procedure. And the method for screening should be cost effective. Now, I have already discussed there are 3 biomarkers which are popular at using in the labs. And out of 3, I would suggest or I would tell them these 2, that capsular antigen based and cell wall antigen based. So these 2 are mostly using the capsular antigen, like this is in case of Cryptococcus fungal infection, Cryptococcus. So this antigen is the polysaccharide. It is an excellent application in case of Cryptococcus infection diagnosis. The best samples are CSF, serum, or urine, and this is basically a latex agglomeration test, and it is also helping the therapy monitoring. And the cryptococcal antigen or lateral flow, and now actually initially it was the cryptola assay where we used to mix up and there was no point of care. But now this is all lateral flow assay or point of care assay. So in that assay, different company they are marketing, like AIIMS. They are marketing, so they have their protocol, and finally if you get the particular band in your strip, it will be positive. Similarly, right now we are using Biocinex CryptoPS, and this is a point of care test. It is very simple. You just put your sample on the strip. It will give the bands accordingly. You can say that whether it is positive or negative. Next biomarker is 1,3-beta-glucan. You know that 1,3-beta-glucan, it is a cell wall property, and it is a trigger to it triggers the innate immunity and activates the coagulation cascade of horseshoe crabs. It is soluble in blood and body fluid, so easily we can detect the beta-D glucan from the blood sample or body fluid. And uh, and this is the available, galactomannan assay. The different Platyella or Aspergillus, Platyella Aspergillus laboratories, and then other laboratories also, marketed the galactomannan assay, but we are using the biodide. So in the colorimetric reaction, we have we are detecting after addition to the substrate. And the EORCT, this is the European Organization of some cancer, and this is the ECIL, the European Organization of some leukemia. They initially determined the cutoff of galactomannan, and initially it was 0.5, but now in our lab we are using the cutoff is 1 and more. Now, up to this, this is the biomarker-based molecular diagnosis. Now, I will start the last part, or the proteomic-based diagnosis, that is MALDI TOF. This is my area. I started this area working on 2014, and MALDI TOF is identification of microorganisms by analyzing ribosomal protein. And it is introduced in the laboratory for rapid and faster identification and accurate identification of bacteria, as well as yeast and mycelium fungi. Now, in India, we have only two commercially available MALDI TOF. We have one is the Bruker, another is the Biomarker. Now, any machine, we should know the principle. The principle of MALDI TOF MS is, this is simple mass spectroscopy, separation of molecules based on the mass to charge ratio. In this MALDI TOF assay, there are 4 steps. One is the sample preparation, which is most critical in case of fungal infection or in case of fungal identification. Then ionization, detection, and data analysis. And this is the schematic representation. This is the MALDI plate. We are, we are putting the or spotting the sample here. Then we put into the MALDI machine, and then laser beam hits this spot. Then it gets ionized. This ion run or travel into the off or time of flight, and this molecule, they travel according to their molecular weight. Hence, the higher molecular weight, it will go slow. Lower molecular weight, it will go fast. And there is, in the end, there is a detector. This detector is detecting a move of this all molecule, and it creates a spectra. So this spectra is like this. And this is the real MALDI TOF. I have just clicked from our MALDI TOF. So here, when you are spotting and you are seeing, this looks like this, and you hit the laser, and this kind of spectra will generate. So now, one thing you have to understand in the MALDI TOF, how we are identifying, because it has a database. So what is database? Database means, let's say, if we talk about Candida albicans or Candida tropicalis. So there are ATCC isolates, there are CBS isolates, or there are, the sequence proved clinical isolates. So the company, what they have done, they collected authentic at least 10 to 15 isolates. They extracted the protein, or they made the spectra like this, and they made a database. And when we are trying to identify our test sample, so our generated spectra, it matched with the database spectra, and it gives a score. The score can be above 2, so 2 to 3, 1.7 to 1.99, and below 1.7. So above 2, it is species specific. 1.7 to 1.99, it is genus specific, and below 1.7, it is no reliable identification. So I was talking to you now the database. So database, there are lots, multiple entries. So when we identified the Candida krusei, this is looks like. So You see, the maximum heat is Candida krusei, RV419. You see, the score is 2.3. Next is CBS isolate, score is 2.0. That way, you will get at least 15 to 20 isolates there in database, and according to your match, it will give you a score, and also it will give you a list and which strain, what score. So that way we create. So database is very, very important in case of MALDI. Let's say in one test isolate you have identified, you could not identify it, and you tried with the MALDI, but you are not getting any identification. It is score is less than 1.7. So what would be your interpretation? One interpretation, it could be that your spectra didn't generate. So there is no proper spectra. Another interpretation is that there is no database. Means if your spectra is not there, so you cannot get the identification. So that's the way you have to be very careful. So what we do when this kind of scenario happened, we immediately do the sequencing of that particular organism, and then we check whether this particular organism it is in the database or not. And if it is not, then we are adding into the database. Now, this is the workflow of a clinical laboratory. The simple overnight growth of yeast. Then you spot it into the MALDI plate. Then add the formic acid and matrix for the on-plate extraction. Then you put it into the machine, and it will give you the identification. It takes about half an hour. So you see how quick it is. So what I was trying to say that it has been found that this ribosomal protein, in case of different species, it is different. So I just wanted to show you in case of Candida albicans, Candida tropicalis, Candida krusei, Candida glabrata. Even if you look at it, but when visually, even in the naked eye, it is different. But if I show you, this is a Candida albicans, different isolate from different patient. You see, this is identical and exactly the same. So that is the beauty of this machine. It is highly, highly accurate, but you have to do it in a proper way. Now, if we talk about the use of MALDI, there are different uses of MALDI, like identification of bacteria, mycobacteria. So definitely this is not my area. But rest 4 area, identification of fungi, yeasts and molds, typing by using MALDI TOF, antimicrobial susceptibility testing, direct testing from clinical samples. In all the area we have worked and we have published paper. Now, the crucial part of this MALDI TOF is identification for fungi. This is the sample preparation. So sample preparation, it can be in 4 ways. One is the direct smear. Most of all the bacteria, they can use the direct smear. Another is the on-plate extraction. Means you have the pure colony, you spot it, then on the top of that spot, after becoming dry, that is the formic acid. And then when it gets dry, you add the matrix, and then you go for identification. So this is called on-plate. Means you are extracting, but it is on-plate. And other 2, that is the organic solved extraction, and then mechanical disruption. Means when we have the mycelial fungi, when we have the black fungi, their cell wall is really tough to break. So in that case, we are using the massive vortexing or vigorous vortexing, or we are using the different mechanical disruption like sonication, vortexing, or sometimes we tried with crushing with liquid nitrogen. So we have so many papers. I would like to touch upon 1 or 2. This is the paper we published in 2015, and the worldwide this paper is cited. So in this paper we have standardized with 354 yeasts, and then we validated with 367. And if you see a green name like Candida auris, Candida Kodamia omeri, and Candida vishwanathai. So I'm talking about this work. It was in 2015, and that time Candida auris was not in the scenario. So there was no Candida auris. And in that time only we have included this Candida auris into the database. So the Bluecard system, this is an open system, and the beauty of this machine is, you can do a lot of manipulation. You can standardize your, the extraction technique. You can create your database, in-house database, without even touching the Bluecard original database. So there are a lot of, relaxation you have with the Bluecard database. Now, just summary of my work. Only one point I want to tell you, that although in the MALDI-based identification in Bluecard, the score, it should be 2 and more for the species level. But in our study, we have found even if it is 1.7 and above, we could confidently say this could be a species level. Now, this technique, it is not only implemented here. We have guided so many other labs. They are using daily in a routine work for the MALDI identification. Now, I just wanted to tell you that the use of MALDI TOF changed our life. Wow! Because if you see, when we have this yeast identification before MALDI, so when we have the positive flag bottle, then we first to do the gram stain. If we have yeast cell, then we are culturing it. If your culture come positive, then you do the you do the germ tube test. You do the a lot of biochemical like sugar assimilation. You do the promagar, so many things. So it used to take couple of days for one yeast identification. But when the MALDI came, if the organism is in your database, it takes only half an hour. That's it. So that's why I made this. So before this, yeast identification, before the MALDI, our situation is like this. Even I can tell you, when we started the MALDI in our routine, during that time, we had around a couple of hundreds sugar assimilation was pending. But now nothing is pending. Every day we are updating. When we started the mold identification by MALDI, it was a big challenge. Why challenge? Because there was lack of standardization of protein extraction protocol. Because in compared to the yeast is really easier. But when we started or tried with the molds, it was not that easy. And also the lack of database. Means the Bluecard database, I'll show you, we have created a nice database. It is far better than Bluecard. So there was not many of the molds, it was in the database. So we published another paper and we standardized it. I just wanted to show you this because in case of yeast, you grow them on the tape and you can use the solid media. But in case of mold, we have seen not only a single method work. It is, fungus to fungus. It is mold to mold. We have to standardize. So whatever the ideal, we have to choose. So in so many cases, we have seen that even the solid culture, we are getting very good identification. But the Aspergillus, we found that 12 to 14 hours growth in the rotosplin is the best. So this is the way we have standardized. And then we have published the paper where we found that our technique, we have tried different technique, and that our technique means extended vortexing with glass beads. And when we, extended our database, we got 100% mold identification. Another paper we have published that is really helpful for the melanized fungi or the back fungi. You know that it's a very slow grower, and some of the cases even you are not getting the sporulation. So one of the of your choice is DNA-based sequencing, and another we have created a good database for this. And now we are, identifying the melanized fungi without any problem. So when we started our work, this was the database of Bluecard of the melanized fungi, but we have enriched or enhanced the database with about more than 30 different species. Now, last and another area what we worked and recently we published, that is the identification or the MALDI-based diagnosis from direct samples. Direct sample is a challenge with the MALDI-based diagnosis because In case of MALDI, we need the organism in certain number, but let's say the tissue sample. If you tissue sample, if you homogenize and extract the protein, I mean, the level of the protein or the spectra you will get, it will not up to the mark. So that's why we thought one of the area where we can work, that is the direct blood sample. So what happened when we, it is the schematic, when we have got the, beeping or beep is there, it means that positive flag bottle, we immediately, what the usual technique is, we go for the overnight culture. But what we did, we took out some blood and we immediately did the gram staining. And in the gram staining, you can easily differentiate whether it's a yeast or it's a bacteria. Then when we got the yeast, we process this blood sample, we make the pellet, and we do the MALDI TOF identification. So you see, this is 18 to 24 hours, and it is only require 1 hour. So how quick you can do this? And this is the different, the blood yeast we have just recorded, because we have seen in our experience that in case of glabrata, if we see this kind of phenomena, because glabrata, they are forming clumps and they live together. So that's why if we see this, we can say it's a glabrata. Candida albicans, if you incubate them a little longer, you will get big hyphae, So this way, we have noticed that the different the different candida, they have different, the appearance in the gram state. We recently published this paper, that is identification and broth microdilation, antifungal susceptibility testing of yeasts directly from the automated blood cultures. The reason behind, if you think that in the regular mycology lab, we are first identifying the yeast, and then when after identification, again you subculture for the pure growth and you do the AFST. So AFST, you put whether it's a broth dilution or distribution, again 24 hours it takes for the result. So we have noticed at minimum of 72 hours it took for identification of the blood-borne candida up to and then their susceptibility up to 72 hours. So we reduced this up to 24 hours. I'll show you. So we have done the direct ID, or we have done the direct identification, and this after direct identification from the pellet, we have also done the AFST. So this is the schematic. When you get a flag bottle, you just do the gram stain, then you take 4 ml of blood, you make the pellet, and this pellet, one portion you go for MALDI identification, and rest of the pellet you process. We have modified and we have developed this process, and then you clean it up, this pellet, and then this pellet you set the inoculum size and you go for the AFST. So you see the AFST for the final direct AFST within 24 hours, but for the identification, it is just more than 1 hour too. So this is really, we are doing it in the routine laboratory now. So when we standardized, we started with different volume of blood, like 2, 4, 6, and 8 ml, and then we spotted different quantity of sample on the MALDI plate. But finally, we got a combination of 4 ml of extracted positive blood culture bottle, and spotting 1 microliter is optimum for our diagnosis. Now, this is the different sensitivity and specificity. I'm not going to go for detail. It's already so much time I have to I'm taken. Recently, we have published another paper, because this was on demand. So when we went to Europe, 1 or 2 group, they were demanding that, sir, although there are so many protocols available for the identification of dermatophytes, but none of them they are working. So what we did, we took all the protocols, we tried for the dermatophytes, and we found that those protocols we modified, and we found the best protocol, and this has been just published. And you see, this we have also created the database here because there was not sufficient database for dermatophytes. Now, I was talking to the database which you have created in our setup. So this is the total blue card existing database, and this is the database we have created. You see, there are 60 different species, and mostly they are molds. So we have created this. Even I asked Karuna, but possibly the Vitek system, it is not possible because they are not compatible. But if any Blueprint they are interested, we can share your database and you can use it. Last slide, this is the another slide. Last slide I compared because my topic was whether it can be this MALDI TOF and then alternate. So here I compared the different techniques like conventional, then DNA based and MALDI. So you see the time 2 to 5 days, 2 to 3 days, 30 minutes. Even if you talk about the money or the cost, conventional 4 to 500, DNA sequence based 750 to 2000, whereas the MALDI is only 5 to 10 rupees. So that's that's the striking point. But the MALDI is really, really good, but there are certain drawbacks also. Like the machine costs very, expensive. It's more than 1 crore. Like now, Bluecar is almost 1.4 crore, and even Biomarker is more than 1 crore. But once you have this machine, if you can, convince your administration, it is a one-time investment. Once you have the machine, the consumable is nothing, the negligible. Very, very easy. Like you have to buy the, acetonitride, and you have to buy the formic acid. That's it. So it is very important if you can convince your, administration, and if you once you get this machine, it is fantastic. And in the end or in the conclusion, I can tell that nucleic acid-based diagnosis of fungal infections are accurate, reliable, and reproducible. PCR-based diagnosis direct from sample, it's still evolving and need more standardization. RT-PCR-based diagnosis, it should be more and more. Right now we are also trying to standardize. And biomarker-based diagnosis, it is best for the Cryptococcus infection. Galactomannan assay and beta-D-glucan assay is also becoming very popular. And of course, MALDI TOF-based diagnosis of fungal infections are getting more and more attention. And lastly, I would like to, introduce my co-faculty. This is Dr. Professor Chakraborty, who is our mentor and the captain of our team. He is Dr. Shiva Prakash. He is now the in charge, and he will be the successor of Dr. Chakraborty. It is myself and Dr. Harsimran, our youngest faculty. She is now the associate professor. And these are the students. They are working with me. Saurabh working with the Candida auris drug resistance. Jamini working the development of rapid diagnostics for fungal keratitis. Brinda is working on biofilm and drug resistance. And Snigdha is working the direct identification by using MALDI for the fungal infection. So that's it. Thank you for your patience. I think it's almost an hour. So
No
I'd be
problem,
very
sir.
happy. I'd be very happy if you have this patience. I have a 7 minutes video which I have uploaded with the request of Dr. David Denning from UK, and now it is in YouTube. I think almost 3,000 viewers is already seen. So if the audience is interested, you can
Yes,
run
sir.
that video.
Yes, sir. You can play, sir.
No, Corona said from why I
Yes, sir. Yes, sir. I have got it. I am going to play it, sir. Yes,
okay,
sir.
okay. No, but screen I am sharing. So shall I?
Okay, sir. No issues.
Okay. So 1 minute. It is not sharing?
Sharing, sir.
It's sharing?
But sound is not there, sir. So shall I play from my end, sir?
Okay, 1 minute.
Sound has to be shared, sir. Okay,
one
sir.
minute.
Yes. Sir, sound has not been shared from your screen
Okay,
share.
1 minute, 1 minute, 1 minute.
Sir. While sharing, sir, there is share sound in the Zoom.
Okay. No. You are not?
No, not audible, sir.
Uh,
Shall I play from here, sir?
yeah, if you can,
Okay.
definitely.
Yes, sir.
Today we are going to demonstrate matrix-assisted laser desorption ionization, time of flight mass spectroscopy, or in short MALDI TOF MS, for the faster and reliable identification of clinically isolated fungi. Friends, this MALDI TOF MS is now being used for routine identification of bacteria and fungi in so many laboratories across the world. Here in PGIMER Chandigarh, India, my group is using this technique for the rapid identification of yeasts and molds since 2014. In this technique, basically the ribosomal protein spectrum is used to identify the organisms. This is the MALDI machine. It is called Biotyper. The Bruker Germany marketing this machine. This machine I am going to demonstrate how we can identify the clinically important fungi. You know that fungi, it can exist either yeast, mold, or yeast and mold in case of dimorphic. But we are going to demonstrate only the identification of yeasts and molds. So if I show this plate, this is a pure yeast growth, and you see this is the isolated single colony, and this is the fresh growth process, these species for the identification. This plate is mold, and this is a fusarium growth, and this is also young growth. We are going to show step by step how we can extract the protein, how we can spot it into the MALDI plate, then put it into the machine, and how we can use this machine to identify the clinically important fungi.
Now show here the identification of yeast by Maldidof. Remember, the yeast to be identified should be a fresh growth, usually 14 to 18 hours. We take 3 to 5 single colonies by using a sterile loop and resuspend it in 1 ml of double distilled water. This is then vortexed to wash the cells and remove any impurities from the media or unwanted proteins which could interfere with the MALDI result. After washing twice, the pellet obtained by centrifugation has to be resuspended in 100 microliter of double distilled water and mixed properly. The MALDI plate can be of two types, either a reusable steel plate as we are using here, or a disposable plastic plate. We add 1 microliter of the yeast suspension to the MALDI plate. Here is a spot close-up image on the MALDI plate. Now we add 1 microliter of formic acid on top of the dried yeast spot as standardized in our laboratory. Finally, we add 1 microliter of matrix on the dried spot before putting it into the MALDI machine for identification. We will now present the identification of mold using Maldidof. For the identification, mold can be grown in either a solid media or a liquid media. Here we are showing a growth in solid media. The growth in solid media should be gently scraped with the help of a L-shaped wire, and this growth must be suspended in a centrifuge tube containing 1 ml of double distilled water. We wash the growth with double distilled water. For growth in liquid media, we can use a rotospin as shown here. This is a picture of growth in liquid media grown on rotospin. We take the growth from the liquid media with the help of a pipette and place it inside a centrifuge tube. This must also be washed just like the solid media growth. After washing with double distilled water, the final wash must be made with ethanol. After the ethanol wash, this centrifuge tube is placed to form a pellet. The pellet must be completely dried by using a vacuum concentrator before proceeding further. This removes any residual ethanol present inside the tube. Now the pellet is dried and it has to be emulsified with 50 to 80 microliter of formic acid, followed by incubation at room temperature for 5 minutes. We will then add glass beads 0.5 mm in size, acid washed. The quantity of glass beads must be equivalent to the volume of the original pellet size. Then we will add the equal volume of acetonitrile as formic acid, that is 50 to 80 microliters. This suspension should then be vortexed vigorously at least for 10 minutes. This ensures proper lysis of the molds. After vortexing, the lysed suspension is centrifuged. Now, after centrifugation, 1 microliter of the clear supernatant is taken and spotted onto the MALDI plate. After drying this spot, 1 microliter of matrix is added on top of this dried spot. Now your MALDI plate is ready. Gently place your MALDI plate into the MALDI biotyper machine as we are showing here. We will also show you here how the plate can be removed from the load port. Grip the plate from both sides and remove gently. Now open MALDI biotyper real-time classification software and click on File. Select New Classification and name the project. Click on New. Select position of target samples and right-click and select Add Analytes. Give the ID to each sample and click on Next. Flex Control automatically acquire the spectra of each sample and results are generated in Biotyper real-time classification window. Then select the database to compare your generated spectra and give the final score. The main advantages of MALDI TOF MS based identification is that it is faster, reliable and cost effective. As per a rough estimation, one yeast or mold identification by MALDI costs 10 cents compared to sequence based identification which costs more than $10. Additionally, it takes only a few minutes for MALDI based identification. These are the publications related to MALDI based identification and detection of resistant isolates. These are 3 faculty members working under the guidance of Dr. Arunalok Chakrabarti. These are the people working with Dr. Anup Ghosh. Thank you.